Okay, the presence of, here see the topic, the presence of rigid, the presence of rigid cell wall does not allow cytokinesis by constriction. See here, here presence of rigid cell wall does not allow the, see here, see here rigid cell wall, this is the rigid cell wall, this rigid cell wall does not allow the cytokinesis by constriction. See, this is the cell membrane. This cell membrane gradually grows and converted into like this way, like this way converted. See here, this is the cell wall, this is the middle lamella. So this is gradually grow and cut off and forms like a cell wall. So due to the presence of this rigid cell wall, it prevents the cell division. It prevents the cytoplasmic division. See here, the presence of rigid cell wall does not allow cytokinesis by constriction. So instead, cytokinase begins by formation of a cell plate in the middle of the cell during late anaphase. The spindle fibers don't disappear at the equatorial plane and form a cell plate and represents the middle lamella between the walls of the two adjacent cells. As the cell plate gradually becomes distinct and develops into new cell wall, it divides the plants into two cells. See here. So instead of this much explanation here, simply see here. So see here, this is the cell membrane. So this is cell membrane is forms gradually thick wall. So it cut off and forms two cell walls in between two cells. It is one nuclei, it is one nuclei. So gradually. So due to the presence of this cell wall, it enables to perform cytokinesis division. So that here one middle lamella is formed. This is called middle lamella. A thick construction is formed. It stops the cytokinesis division. And finally, so two cell walls are formed. Two cell walls are formed. In plant cells like this way, in animal cell like this way. Okay. So this is called cell wall. This is cell wall. This is cell wall. This is the cell wall. This cell wall divides the one cell into two cells. Finally, as a result of mitosis in plant cells, two cells are formed. So these two cells also formed from single cell prophase. It is divided into two cells. Finally, two daughter cells are formed. This is called mitosis. Okay, that's right. Here see, instead cytokinesis begins by formation of cell plate in the middle of the cell during late anaphase. Actually, this starts in plant cells. Anaphase onwards, anaphase from this third phase onwards, formation of two cells formed in plant cells. Okay, so instead of cytokinesis begins by the formation of cell plate in the middle of the cell during late anaphase. Okay, that means what, from anaphase onwards only. So middle lamella formation slowly starts. The spindle fibers don't disappear at the equatorial plane and form the cell plate that represents the middle lamella between the walls of the two adjacent cells. This is only. So this cell plate represents the middle lamella in between two adjacent cells. So as the cell plate gradually becomes distinct and develops into new cell wall, it divides the plant cell into two. So finally, as a result of mitosis in plant cells and animal cells, so two cells are formed. Next, here one more topic: formation of syncytium. Formation of syncytium. See here, syncytium formation. See, in some organisms, karyokinesis is not followed by cytokinesis. In some organisms, So generally, most of the organisms karyokinesis are after immediately cytokinesis is formed. So after finishing cytokinesis, two daughter cells are formed. If cytokinesis does not take place, cytoplasm if does not divide, two daughter cells are not formed. Okay, in some organisms, karyokinesis is not followed by cytokinesis. That means prophase, anaphase, prophase, metaphase, anaphase, telophase. After these, cytokinesis does not take place. As a result of which, multinucleate condition arises. If cytokinesis does not followed by karyokinesis, then what happens? Multinucleate condition, number of nucleate condition is arised. That condition called syncytium. Example, liquid endosperm in coconut. In coconut plant, cytokinesis divisions does not take place. As a result of that, number of nuclei are formed. Example, liquid endosperm coconut. So liquid endosperm means what? Coconut water. Actually, coconut water does not show. Coconut plant does not show cytoplasmic division. Cytokinesis does not take place. If cytokinesis does not take place after the karyokinesis, multinuclear number of nuclei are formed. That is called syncytium. Formation of multinuclei is called syncytium. Example, liquid endosperm. Endosperm means what? Liquid endosperm means what? Liquid water in coconut. If you observe the coconut water, it has multinucleus, syncytium. Okay, ma? That's all. Next topic. What is the importance of mitosis? So, significance of mitokines. Significance nothing but importance. Importance of mitosis. See here, if you will see identical gene composition cell, identical gene composition, identical gene composition. Gene composition is identical. The cells contain same number of chromosomes as the parental cell. That only in parental cells, in daughter cells, gene composition is identical. Cells the parental carry the same hereditary information as in parental cells. See here. The cells carry the same parent. See here. The cells carry. Here point is what? The cells carry the cells carry the same hereditary information. Here, little bit spelling mistake is there. The cells here, the cells carry the same hereditary information as the parental cells. Okay, that only I am saying it is prophase. In prophase, 46 chromosomes will be there. As a result of mitosis divisions, two daughter cells are formed. Here also 46 chromosomes, here also 46 chromosomes, because mitosis is the equational division. Daughter cells will have the same number of chromosomes as their parental cells. There is no variations in genetic information, that only. So in parents, whatever may be the parental cells chromosomal composition is there, the same chromosomal genetic composition is present in the daughter cells also. It gives genetic stability with population. So mitosis divisions will give genetic stability within a population. Genetic stability means what? So in huge number of population, if you will observe, after mitosis, genetic stability will be there. Why? Mitosis division takes place in somatic cells. In somatic cells, so here mitosis division takes place. Here, during accident, some tissues are cut off. In that place, due to mitosis, new tissues are formed. So, before the what chromosomal number is the same chromosomal number is maintained. In such a way, if you observe after mitosis divisions in a group of population, genetic composition is same, 46 chromosomes. DNA contains same adenine, guanine, cytosine, thymine. In that any changes does not take place. That is called identical gene composition. Identical gene composition. Next, growth. The increase in number of cells bring about body growth and scope for cell differentiation. That only as a result of mitosis division. Already we have discussed it. So meristematic tissue undergoes rapid mitotic divisions. As a result of mitotic divisions, it gives rise to. As a result of mitotic divisions, it gives rise to new cells. As a result of formation of new cells, it undergoes body growth. It brings about body growth and scope for Differentiation. Differentiation means what? Millions of cells are formed. Those are all the cells are differentiated into roots, stems, leaves, fruits, flowers, seeds, etc. That is called growth. Next, third point, cell replacement and repair. See, the replacement of lost cells like skin cells. See, skin cells, blood cells, and cells of digestive tract and wound healing occur to varying degrees. See, after bathing, we will take towel and we will wipe our skin. We will wipe our skin with a towel. At that time, some dead cells peel off. Some dead cells, old and worn out cells are peel off. And new cells are formed daily. 10 into 10 power 24. 10 into 10 power 24 new cells are destroyed and same number of new cells are old cells are destroyed and same number of new cells are formed. See, skin cells daily, skin cells are destroyed and new cells are formed. Blood cells, the red blood cells white lifespan is 120 days. After 120 days, new cells are formed. So white blood cells lifespan is 12 to 13 days. After 13 days, new white blood cells are formed. In such a way, cells of the digestive tract. Wound healing occurs in varying degrees. That entire process depends upon mitosis divisions. The cells of the upper layers of epidermis from the lining of the digestive gut, the digestive system, blood are constantly replaced. Not only in our body. So in some of the organs, old cells are destroyed and new cells are continuously formed. Okay. In such a way, some cells are helpful. Next, here, regeneration and asexual reproduction. In some animals, the regeneration of the last part leading to a mode of multiplication. See, regeneration, already fragmentation we discussed in first chapter. So when the body is divided into fragments, so those fragments are undergoes mitosis divisions. Mitosis is nothing but here asexual reproduction and forms new cells. Whatever maybe the parts are lost, those parts are again regeneration. So that process commonly takes place in planaria, sponges, etc. In those organisms, regeneration process occurs. So in them, regeneration is a type of asexual reproduction. In plants, asexual reproduction is made of vegetative reproduction. So in plants, asexual reproduction nothing but vegetative reproduction, by which the number of species increases. Yes. So by vegetative propagation, we can increase the number of new plants. For example, you observe one jasmine flower plant or hibiscus plant or rose flower plant. Some you cut it and keep it in the soil and during rainy seasons, it will develop into new plant. So that is a kind of a vegetative reproduction. That's all. Vegetative reproduction means what? In that vegetative, the plants which are showing vegetative reproduction, in them mitosis divisions taking place. As a result of mitosis divisions, new cells are formed. Those new cells leads to the formation of a new plant. So mitotic divisions in the meristematic tissue like apical meristem, lateral cambium result in a continuous growth of the plant throughout their life. Already we discussed this point. So as a result of mitotic divisions in apical meristem, lateral meristem, root apical meristem, shoot apical meristem, undergoes rapid divisions and produces new cells. As a result of that, growth is occurs throughout the life of the plants. Uncontrolled mitotic division may give rise to tumors of cancers growth in the body. Yes, one important point. So cell division, either mitosis or meiosis, these are the divisions are highly controlled and coordinated divisions. When our body needed, at that time only mitosis divisions takes place. Remaining times, so mitosis divisions are a highly controlled process. But in some of the organs, this controlling mechanism is failed. Without controlling and coordination, the cell division is continuously divides, redivides and produces millions of cells. Those many of cells are turned to be tumors of cancers. Okay, that's all. So tumors of cancers growth in the body. Okay, that's all. Okay, so those tumors leads to the cancer. Cancer is of two types. Malignancy cancer, benign cancer. Benign cancer through surgery we can remove. Malignancy cancer leads to death. About cancer, more and more information, second PUC will study. Okay, ma? This is the explanation about mitosis. Okay, so it is very easy topic, ma. Nothing difficulty. So once with the help of this knowledge, if you read, you will get to know that Okay, if any doubts are there in our regular classes, you can clarify. Okay, so second PUC, these are all again briefly we will revise for objective discussion. Okay, that's that. Next, with this successfully, mitosis is finished. Next, meiosis. See ma here, meiosis, meiosis divisions. Okay, students, are you getting my voice clearly, ma? Are you getting my voice clearly? Is there any disturbance? Okay, thank you. Next, let's start next topic. Meiosis. Meiosis occurs in male and female reproductive or germ cells. Already I am saying this is meiosis divisions always occurs in male and female reproductive germ cells. That's maybe testes, ovary. In testes and ovary, generally meiosis division takes place. Other than testes and ovary, remaining organs, mitosis divisions takes place. That's why in mitosis divisions, chromosomal complement, genetic complement is always constant. It occurs during gametogenesis in plants and animals. So whenever meiosis divisions does not take place, for example, children are there up to 12 years, up to 12 years or up to 11 years, up to puberty stage in their body, only mitosis divisions takes place. Meiosis divisions are not started. When they are reached to puberty, that is, that means menarche stage. Puberty nothing but menarche. When the boy girl reach to puberty stage, at that time only in their body gametogenesis takes place. Puberty stage only gametogenesis takes place in plants and animals. Both in plants and animals during gametogenesis only meiosis divisions starts. Okay, ma? That's all. Next here, during meiosis, a diploid cell, see, a diploid cell undergoes two successive divisions. Meiosis 1 and meiosis 2. As a result of meiosis 1 and meiosis 2 divisions, it gives rise to 4 haploid daughter cells. Yes, 4 haploid daughter cells. In males, 4 haploid daughter cells. One time, if meiosis division is finished, four sperms are formed. 4 sperms. These 4 sperms are matured. In females, same process takes place. Only one ova is released. In males, 4 sperms are formed. In males, 4 sperms are formed. In females, only one ova is formed. So how it is takes place? See, diploid cell undergoes meiosis 1 and meiosis 2. 4 sperms are formed. Only one ova is formed. Okay, that's all. Here you have to remember, one cell gives rise to one diploid cell gives rise to 4 sperms. In such a way, millions of gametes are formed. Millions of sperms are formed in male. Whereas in female, only one ova is formed. The daughter cells so formed are called gametes. Those gametes are in case of male persons, male gametes sperm, female gametes called ova. In female persons, female gametes, those are called ova. Okay, the gametes contain only half the number of original chromosomes. That only I am saying to you many times. Gametes, diploid cell undergoes meiosis divisions and forms haploid sperm, haploid ova. What is the reason of forming a haploid sperm? Means already I said many times. So it contains 23. These two together when fused forms 46 chromosome zygote. That's why here 23. Sorry, diploid means 46 chromosomes became 23 chromosomes. So to maintain chromosomal number constant, meiosis divisions are needed. If any person's meiosis divisions does not take place, so then what happens? In male persons, 46 chromosome sperms formed. In female persons, 46 chromosome ova formed. If these two are fertilized, zygote formed, 93 chromosomed baby formed. So 93 chromosomed baby is abnormal baby. They will face number of health problems. So that here the importance of meiosis is this is when chromosomal number is maintained half, then 46 baby forms. 46 chromosomed zygote is formed. That's why meiosis division is also called reductional division. The gametes contains only half the number of original chromosomes. Half the number of original chromosomes. The meiosis is also called reductional division because the number of chromosomes in the daughter cells gets reduced half. See the diploid parent body. For example, my body is diploid parent body. Okay, like that. In gametes, haploid sperm, haploid ova. When these two are combined with each other during intercourse, 46 chromosomes, baby formed. Okay, that's so from diploid to haploid. It's called diploid parent body to haploid gametes. Meiosis involves single duplication of chromosomes followed by two successive divisions. So meiosis 1 and meiosis 2 divisions. Generally here meiosis divisions always takes place in two types. Meiosis 1 here, meiosis 1, meiosis 2. As a result of meiosis 1 and meiosis 2, sperms and ova are formed in human beings. Okay, so meiosis involves a single duplication of chromosomes. In this one duplication, that continuation duplication takes place here by two successive sequential divisions, that is meiosis 1 and meiosis 2. First of all, let us see meiosis 1. Meiosis 1. Various stages of meiosis. Meiosis is divided into prophase 1, metaphase 1, anaphase 1, telophase. Parallelly, cytokinesis 1. Prophase 1, meiosis is divided into 5 types. Prophase 1, metaphase 1, anaphase 1, telophase 1. It is easy, ma. If you learn perfectly mitosis, that only here continues. Same points here repeated. A little variations. Prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis 1. Next, based on the chromosomal behavior, prophase 1 again divided into subdivide. Again, prophase 1 subdivided into following 5 phases. So these are leptotene, zygote, pachytene, diplotin, diakinesis. In mitosis, we have studied prophase, metaphase, anaphase, telophase, cytokinesis. Same here, you will write prophase 1, metaphase 1, prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis 1. Depends upon chromosomal behavior in different stages. Prophase 1 again divided into 5 subdivisions. Leptotene, zygote, pachytene, diplotin, and diakinesis. Here the names given, see, leptotene, zygote, pachytene, diplotin, and diakinesis. Okay, ma? That's it. Now again, one by one we will see. First, leptotene. So leptotene. See here, leptotene. See ma here, leptotene. This is the leptotene diagram. In this one, leptotene diagram. In this one, see here, chromosomes shortened. See, these are the chromosomes actually. These are all chromosomes. Chromosomes are shortened. Chromosomes are shortened. Chromosomes are shortened and are visible as a single strands under light microscopes. These are all visible as a single strand, like this way, single strand, like this way, single strand. Single strands in light microscope. Next, beaded appearance of chromosomes due to chromomeres. Beaded appearance of chromosomes. Beaded appearance of chromosomes. Beaded means what? See, in olden days you might have seen 5 years, 2 years back, 3, 4 years back, pearl chain. So pearl chain like this only will be there. These are called pearl chain like this way. Okay, Like this. Beaded means these are all called beaded. Beaded appearance of chromosomes. These chromosomes are seems to be like this way due to chromomers. Chromomers nothing but like this way, like this. These structures called chromomers. Due to the chromomers, the chromosomes appears to be chain-like structure. Chain. Just you remember pearl chain, white pearl chain. How like that chromosomes are appeared in leptotin. So nuclear membrane and nucleoli intact. This is the nuclear membrane. This is the nucleoli is intact. Intact means complete structure. So compaction or condensation of chromosomes continues throughout the leptotin. Compaction nothing but conduction. Conduction means so like this way. Here two only shown like this way. Conduction means thick structures are formed throughout the leptotin. Okay, that is the explanation about leptotin. Leptotin. So that you have to remember. Next point. Next one is zygote. Zygote. Next one what? Zygote. See here, zygote. Zygote diagram here given. That diagram I will show here. See here, in the zygote, homologous chromosomes, that is maternal chromosomes and paternal chromosomes are paired up. Maternal chromosomes and paternal chromosomes means what? In my body, from my father 23 chromosomes, from my mother 23 chromosomes. These chromosomes starts pair up. See, 23 chromosomes from my father, 23 chromosomes from my mother. So those homologous chromosomes from this 23 pairs, from this one chromosome starts pairing like this way. Okay, this is one chromosome. This half from father, remaining half from mother. In my body, reproductive organs from my father chromosomes, mother chromosomes comes and starts pairing. That is called homologous chromosomes. Maternal, paternal chromosomes pair up. Pair up means this is homologous chromosomes. In the homologous chromosomes, this is comes from father. This is comes from mother. Pair up. Paired homologous chromosomes seen as bivalent or tetrad. Bivalent. 1, 2. Bivalent. Or these are also called tetrad. 1, 2, 3, 4. Tetrad. Pairing process called synapsis. This pairing is called synapsis. The pairing of homologous chromosomes are called synapsis. So chromosome synapsis is accompanied by the formation of complex structure called synaptonymal complex. So chromosomal synapsis means what? Homologous chromosomes starts pairing. That is accompanied by the formation of complex structure. This is also called complex structure. This is also called synaptonymal complex. That means what? Homologous chromosomes from the paternal maternal starts pairing. That is called synapsis. So that synapsis is also called synaptonymal complex. Synaptonymal complex. Okay, so that's all. Thickening and shortening of chromosomes. Thickening and shortening of chromosomes. See here. So chromosomes like this way. Thick and shortening of chromosomes. Nuclei tend to disappear. Nucleoli means what? So this is called nucleoli. This nucleoli is gradually disappear. Leptotin and zygotin are of short duration as compared to next stages, that is vacuum. Okay, that's all. So leptotin and zygotin both are less time is required. Okay, see here the diagram I will show here. See, so what I said, homologous chromosomes. These homologous chromosomes. So this is white, this is black given, this is from mother, this is from father. This is a nucleolus. So this is called bivalent. This is also called synaptonymal complex. So this is centromere. Homologous chromosomes. So see here, before starting cell division, centrioles are close together. When these are involved in cell division, these two centrioles, these centrioles slowly moving towards opposite pole. This is completely comes to opposite pole. This is called zygote. Zygote. Okay, ma? That's all. Next stage is packaging. Next stage is packaging. So the word itself saying the meaning packaging. Packaging. Okay, ma? See, packaging. So packaging means what here? Packaging. This stage is characterized by the appearance of recombinant nodules. See here, like this way. Pack nothing but packaging. Like this chromosomes are starts packing. See, packing. In this one, homologous chromosomes are there. Before the picture, what I shown here, before the picture like this way I shown, na? That is what here will be appeared. So one I will show like this way. One type of pair is black, one type of pair is red. So that is called homologous chromosomes starts, and this is characterized by the appearance of recombinant nodules. Some places recombinant nodules are formed. Recombinant nodules, recombinant nodules are formed. Recombinant nodules are formed. The site at which crossing over. So where the recombinant nodules are there, at this place, another pair, same type of homologous chromosomes comes and attached this. That place is called crossing over. See, at which means where is the recombinant nodules are there, at that places crossing over. Crossing over means what? Like this way. This is the recombinant nodule. At this recombinant nodule, crossing over takes place between non-sister chromatids of homologous chromosomes. You have to remember, it is very important bit. Non-sister. So these two are sister chromatids. These two are sister chromatids. So these chromosomes goes and binds with this chromosome. This will come. This chromosome will come and combine with this. This is called crossing over. Here recombinant nodule will be there. So in between these two are sister. These two are sister. This is to this non-sister. In between non-sister chromatids. These are called individual. This is homologous chromosomes. Individually, these are called chromatids. See here, crossing over occurs between non-sister chromatids of homologous chromosomes. See, homologous chromosomes. See here, I am showing like this way. Okay, it is. Here, I am showing. See here, it is. It will come unattached. Here, this is will come and attach. Okay, here commonly chromatid commonly will be there. This is called crossing over. Crossing over. See, so this is a sister. This is chromatid. This is chromatid. This is chromatid. This is chromatid. Chromatid. So this is a sister chromatids. These are sister chromatids. This is to this relation is non-sister chromatids. Non-sister chromatids of homologous pairs. These are all homologous pairs. So this pair is from mother. This pair is from father. This pair is from mother. This pair is from father. Same. In between non-sister chromatids of homologous chromosomes, crossing over occurs. This is called, see, actually this is like this way occurred, but their pictures we cannot differentiate like that. In this one, how happened? Like this way happened. See, crossing over in homologous chromosomes. Crossing over means what? Like this way. So chromosomes of homologous pair, bivalent chromosomes, become twisted spirally around each other. They appear as a tetrad. See, like this way, twisted. Like this way means what? Like this way, twisted. It is one homologous chromosomes and another homologous chromosomes are twisted. Those are all together appeared as a bivalent. It is 1, 2, or else 1, 2, 3, 4. However, we can bivalent or tetrad. So they are called bivalent chromosome or they appear as a tetrad. Crossing over occurs. Crossing over is the exchange of genetic material between the two homologous chromosomes. Crossing over means what? During the crossing over, what happened? So in this one, in this chromosome, whatever maybe the genetic material DNA is there, that is exchanged with this. In between the non-sister chromosomes, genetic material is exchanged. That is called recombination. The word only saying the meaning. During crossing over, what happens? Recombination. Recombination, nothing but genetic material is exchanged. Genetic material is exchanged. See, crossing over is the exchange of genetic material between the two homologous chromosomes. These two are homologous chromosomes. Crossing over is an enzyme-mediated process and the enzyme involves. So, this is enzyme-mediated process. The crossing over is induced by the enzyme. That enzyme is recombinase. Recombinase enzyme. Exchange of corresponding chromatid segments. This is segment are exchanged automatically. Genetic material also exchanged. Nucleoli is disappear. In this one, dark staining body is called nucleoli. That nucleoli is disappear. That is the explanation about this topic. Packeting is finished. Next evening class, diplotin, we will see. By sharp 3 o'clock, you log in. Thank you.